Welcome to QIAGEN's webinar today about characterizing the microbiome of neonates and infants to explore associations with health and disease. This webinar will focus on the acquisition and development of the preterm gut microbiome from birth and following discharge from intensive care. And specifically, we will discuss the association of the gut microbiome with necrotizing enterocolitis and late onset sepsis, as well as the impact of birth mode. And we will also discuss analysis of multi-omic datasets, including the analysis of the airway microbiome and metabolome in infants hospitalized with bronchiolitis. And it's a great honor to have you all today. My name is Deepak, and I'll be the host of today's webinar. Today, I would like to introduce Dr. Christopher Stewart. And this webinar is focused on characterizing the microbiome of neonates and infants to explore associations with health and disease. And it's a great honor today to introduce Dr. Christopher Stewart to you all. And Dr. Stewart did his PhD from Northumbria University, Newcastle upon Tyne from 2010 to 2014. And later he moved to the US for his postdoc. And currently he is a postdoc researcher at Baylor College of Medicine, Houston, Texas. His research interests are focused on the diversity and ecology of microbial communities and host responses in clinical samples and ex vivo model systems to determine the microbial host interaction in health and disease. And Dr. Stewart is the winner of 2015 QIAGEN Microbiome Awards, and the awards for 2017 are currently accepting applications. You can find more information towards the end of the webinar at the end slide. And I would like to welcome from my side Dr. Stewart, and thank you for joining us today. And it's my pleasure to hand it over to you now.
Okay, thank you. And hello, everyone. Thank you for tuning in to this webinar hosted by QIAGEN today. So I'll be talking about 4 principal areas throughout this agenda. And I'll give a little bit of an introduction to microbiome research and specifically how we conduct that at Baylor College of Medicine here in Houston, Texas. And then I'll discuss a little bit about the normal microbiome development through infancy, specifically in term infants. And then I'll move that into the preterm field, which is primarily where my research is based. And then finally close by talking about the influence of the microbiome in health and disease of preterm neonates. And so Baylor College of Medicine is housed right in the center here, which is right in the heart of the Texas Medical Center. You can see here is Houston downtown, so that gives you a sense of the scale of the medical center itself. And so this is really just a collection of hospitals and research institutions with a great wealth of staff, making this a really fantastic place to do translational research such as microbiome science. And this is from the department of the Alkek Center for Metagenomics and Microbiome Research, which is where I'm based. This is one of our kind of experimental design overviews, and this is pretty common for microbiome science. So for those unfamiliar with microbiome science as a whole, I'll just take you through a kind of schematic about how we would go about conducting an experiment in this area. And so typically you'll have an observed phenotype in a human population, for example, or you can administer that phenotype to an animal experiment. Or maybe as you're looking at a large-scale trial or administering a new therapeutic, and in some cases we know that these trials are very effective, in other cases they may not be. And so by looking at the microbiome, you may be able to ascertain as to why some patients responded expectedly and others seem for the treatment not to work. An example about how we go about doing this then is sequencing is probably the most common means now of interrogating microbiome. And the 2 main principal ways of doing that are using amplicon-based sequencing, such as 16S, so you target a very specific region of a bacteria that's universal across all organisms and allows you to get a sense or survey what bacteria are present in a given sample. Or more recently, due to reducing costs, we can apply whole genome metagenomic shotgun sequencing, and that will give you not only bacteria but other microorganisms like virus, fungi, and archaea. It also allows you, by doing this more powerful sequencing, to get at the species level resolution of the community and also ascertain a lot more about the metabolic potential of that community itself. And so once you've got your data set, primarily you're interested in looking for common or distinguishing features. And then often you're required to go away and do some identification or isolation of specific microorganisms. And so here's a few examples here, and one that's being developed in our lab currently is this electrophoresis technology. And so this is being developed by Dr. Lorenzo D'Amico, who's an engineer in our lab. And this technology allows us to use microfluidics and other apparatus to electrophoretically separate microorganisms. And that can be from blood, for example. If we want to collect the viable cells from a patient with sepsis, we can collect the bacterial cells whilst washing through the whole cell, so enrich for the microbiome. We can also trap specific organisms that we're interested in whilst allowing other bacteria to elute. And much like an LC-based system, we can run this in profiling and look for peaks and bin separate bacteria based on their electrophoretic properties or structural properties. And then once you've got a sense of what your data is showing, often the next stage is then to model that data. And so an ex vivo cell culture model is typically applied, so that could be a cake or cell line. More recently, we see the emergence of enteroid technology, and this is an area that's fascinating me a lot at the moment. So enteroids are where you would take a resection from a patient, you can isolate the stem cells or isolate the crypts and allow the stem cells to regenerate that tissue in an ex vivo model. And so that then phenotypically represents the area of the intestine it was taken from, but then also has the same exposure and genetic predisposition to the certain disease that you're interested in. And you can apply various techniques to test this, such as co-culture modeling. And then more commonly, I think in microbiome science, we see mice and germ-free animals used a lot. And a couple of other techniques that have been applied a lot at Baylor College of Medicine is these bioreactors, and these are being developed in the lab of Dr. Robert Britton, as well as more basic models to explore host and microbiome interactions, such as the C. elegans worm. And Dr. Buck Samuel's lab is doing some fantastic work in this field at Baylor College of Medicine. And then ultimately, through all this, you'll hopefully derive a target, be that taxa, genes, or metabolites. You may have a target for a therapeutic that you feel is going to work. So, for example, administering a probiotic, certain beneficial bacteria to a patient to allow those taxa to colonize and hopefully reverse some of the disease phenotype. It may be metabolites or taxa that are increased prior to disease as a biomarker for disease. And that's really where this technology is allowing us to go in these clinical samples. And again, because we probably have people tuning in here who aren't familiar with the processes of actually going through to data generation and analyses, you'll typically start with your primary sample. So for a lot of my research, this is a biological sample. And then you'll be required to extract DNA or RNA, depending on what you're looking at exactly. Typically, the kit I tend to use for this and have done for a while now is formerly the MoBio, now QIAGEN Power Lysa Power Soil kit. I'd say this is one of the more common kits for DNA extraction, but it's important to note that if you use different kits, then you're going to yield different data. So if you want to compare your data set against an already existing data set, it's important that you follow a similar methodology. And then again, there's variations as to the way we do this, but most typically I'd say we would target the V4 region of the 16S gene, amplify that using the 2x250 aluminum MiSeq paired end based kit. And then ultimately you get a lot of data, and you're required then to filter some of that data to remove the poor quality reads, to make sure that you're only left with low error reads, and then ultimately align those to the database. And this gives you an idea of the steps involved in the pipeline within the CMMR of Baylor College of Medicine. And ultimately we use the Silver database as our reference for identity. And then you get to the fun bit where you can actually start to do your analysis, and that can be things like alpha diversity, beta diversity, taxonomic abundances, and other things, which I'll talk about throughout the rest of the webinar. So just to give you a little bit of background then about some of the existing data in this field, and before I do, just to talk a little bit about why the microbiome is important. So it has a huge range of importance within the human host. Indeed, humans really couldn't exist without their microbiome. And just to draw your attention to a couple of feats here, particularly that I'm interested in around the preterm neonates, which is the protection against epithelial injury and resistance to pathogens. So colonization by potentially beneficial bacteria have actually been shown to promote gut maturation and barrier integrity and tight junction formation, which is important for ensuring that bacteria can't translocate or potentially release the pro-inflammatory response that we see in a lot of preterm diseases that I'll talk about towards the end of the talk. And also by occupying distinct niches within the gut, they can also infer resistance to other potential pathogenic bacteria to come in and colonize. And then indeed, as we know, the more common ones, which I won't really focus on today, but the breaking down of food components and biosynthesis of vitamins and other amino acids are clearly also highly important to be derived from bacteria living within the gut. So here's just a schematic that I published with some friends and colleagues at Baylor College of Medicine last year. And it just shows that through time, the diversity of the microbiome generally increases, but a lot of this increase occurs during the infancy stage. A lot of my talk today will be really focusing on this very early neonatal stage, and then a small part also in the later infancy stage as well. There are several factors which are going to influence the microbiome development through this time. There's some evidence to suggest that there may be some colonization of the infant prior to delivery, so in utero as a fetus. This has only been done at the molecular level, so using DNA, so it's a highly contentious area. There's very little evidence to suggest in normal, healthy pregnant mothers that we can culture bacteria from things like the placenta and the amniotic fluid, but there does seem to be evidence, at least, of microbial DNA in those samples. But I should note as well, as I touched on earlier, with the kids having an effect, when designing a microbiome experiment, it's also important to incorporate controls. And so in a lot of these studies, like the placenta microbiome, the taxa match with what we'd expect to see in kit-negative controls. So even kits have their own inherent microbiome. We just need to be careful that when we do these experiments with low biomass samples, that we account for that fact. And then I'll talk a little bit about delivery mode in the coming slides, and then towards the end of the talk a little bit more about gestational age and the effect of the neonatal intensive care, or NICU exposures. And then probably the biggest factor in contribution to the microbiome development during early infancy is whether you're breast or formula fed. And indeed, breastfeeding has been shown, and I'll talk a little bit about this, to suppress the maturation of the gut microbiome. But when you're receiving breast milk, you have this huge increase in the amount of Bacteroides and, to a lesser degree, lactobacillus that you're colonized by. But then upon stopping being breastfed, the maturation of the gut microbiome really begins. So even if you're being introduced to solid foods but are still receiving breast milk, we still see this somewhat suppression of the maturation. It's really the cessation of breastfeeding that starts to drive a lot of the maturation that we see, where you'll see the microbiome increase in the amount of Firmicutes, particularly your Lactobacillus and your Ruminococciaceae families, and that's kind of a hallmark of an adult-like microbiome. So 2 fantastic papers which came out last year around this time in Science Translational Medicine. The cohort size was relatively small in terms of participants, but the longitudinal aspect of these studies were really fantastic. And so you can see here a study on the left sampled monthly until 2 years of life, and a study on the right sampled monthly until 3 years of life. And they showed very similar findings, and that is that in the early phase of life, your Enterobacteriaceae, or your proteobacteria, reduce pretty exponentially throughout the first 12 months, as well as also the Bifidobacteriaceae, specifically just Bifidobacterium, tend to decrease. And again, that really correlates with just a decreased exposure of breast milk in these populations during that time. And then other genera that we see starting to increase are things like Lactobacillus, which actually interestingly peak around a year of life and then drop off slightly, but do remain relatively abundant throughout the rest of sampling. And then the Ruminococciaceae, which does seem to increase fairly consistently throughout the whole of the sampling. And then these studies also assess differences in the birth mode. And so here within the first 6 months of life, you can see that the Bacteroides here by the dark blue is clearly increased in vaginal infants compared to cesarean, and that's regardless of whether the infant was breast or formula fed. And indeed, this finding was echoed in the Yasser et al. Paper, where they found that the typical vaginal birth pattern has this large amount of Bacteroides all through sampling, even in the first year of life, whereas they categorized the cesarean section infants as having just a lack of this Bacteroides colonization. And then interestingly, this paper also noted that around 20% of vaginal births also seem to have the cesarean phenotype, and that they lack this Bacteroides colonization in year 1 of life. And looking through their metadata, and there was no reason why they could find that this was occurring. For example, it wasn't because of any feeding practices or antibiotic practices or anything like that. So this is something that's really interesting, and will no doubt be examined further in subsequent follow studies of larger cohorts. But then those findings were recently challenged in a paper published in Nature Medicine by some colleagues at Baylor College of Medicine, and indeed a very good friend of mine, Derek Chu, and his supervisor, Kirsty Agard, sampled different body sites of the infant following birth, and also the maternal samples. And it was a really nice study, but I won't go too much into the different sampling, focusing mostly on the birth mode aspect of the study. They found that immediately following birth, there was a clear difference between cesarean and vaginal delivered infants across different body sites. However, 4 to 6 weeks after delivery, they failed to see that same effect of delivery mode. And so there's just a couple of things to note here, that the cohort tested at 4 to 6 weeks, where they failed to find a difference, was much smaller than the cohort that they were exploring immediately following birth. And also, whilst it wasn't significant by the 0.05 threshold of significance that we typically apply to these types of studies, it's clearly trending towards significance. And so it does seem to be some effective delivery mode even sustained after this time. And what would be very interesting is in this cohort to keep following them up to see if in later time points the lack of significance became greater or situated around the significance threshold. So this is still obviously very contentious and open to debate, and I'm sure some future studies in larger cohorts will allow us to get more at this. And one thing to note, that a lot of these studies, in fact all of these studies, were based within single locations, and so what's probably needed is really a multi-geographical study design to really be able to apply these findings to the general population. And the reason I talk about this, the reason that it's important, is because we know that cesarean section increases the later life risk of disease. And this is now, I'm just showing here one epi study, but there's a lot of epidemiological evidence that cesarean delivery increases the risk of several childhood diseases. And a lot of this now is being thought to be in part played by the early development of the microbiome and potential differences there between the microbiome of vaginally delivered infants and cesarean infants during the initial stages of life, and how the immune crosstalk develops because of that. And so here there's just an example of things like asthma and inflammatory bowel disease, where you seem to have a much greater risk of these diseases if you're delivered by cesarean section. And then equally, breastfeeding, as I touched on, is another, if not the key modulator really of gut microbiome maturation, and really shapes the microbiome to move away from that dominance of bifidobacterium lactobacillus into colonization by more taxa from within the Firmicutes phylum. And so this is a recent study published in JAMA by Parinaj, who showed that, and this is actually a novel approach because they looked at the proportion of breast milk in the diet rather than just if the infants were exclusively breastfed or received some breastfed or were not breastfed. And so they found, as you probably expect, that if you get over 75% rather than under 75% of your diet through breast milk, that you have a greater contribution to the infant gut in the over 75% breastfed infants. So breast milk microbiome represent a bigger contribution to the infant gut in those samples taken from infants breastfed greater than 75% of their diet through breast milk. And equally, following the introduction of solid food, if you were receiving solid foods for more than 4 months, you can see that you have this increased relative microbiota maturity. And if you compare that to the samples that were not introduced to any formula, you can see that there's a much bigger delay in the relative microbiome maturation. And as I've touched on, this is likely administered through the lack of Bacteroides after you start to introduce solid food, coupled to the increase in the Firmicutes. And this is also supported by several papers, most notably a 2015 Cell Host and Microbes paper by Frederick Backhead et al., which also found that cessation of breastfeeding really drove maturation of the microbiome. And again, breastfeeding is important not only from a microbiome standpoint, but from these epidemiological evidences that suggest that if you receive breast milk, then you have a reduced risk of asthma. And this analysis showed, which was a meta-analysis of 117 different studies, showed that if you receive any, it doesn't necessarily have to be exclusive breastfeeding, you seem to have this protective effect. And also the longer that you receive breast milk for, the lower the risk of asthma was. And this was also seen across a range of different conditions. And here I'm just presenting another epi study of over 15,000 children aged 9 to 14 years old, and looking at obesity. And just to draw your attention here, that if you received breast milk as an infant, that you were at lower risk of developing obesity at 9 to 14 years of age. And indeed, the longer that you receive breast milk for, the lower your risk of being obese between 9 and 14 years of age was. And then this really plays in with the WHO guidelines of recommending breastfeeding for the first 12 months of life. Okay, and just moving into the preterm space, which is, like I say, where a lot of my research is focused. The preterm microbiome is potentially altered due to a range of factors. For example, we have an increased cesarean section rate in this population. Also, preterm infants have limited environmental exposure. So following birth, they are housed in sterile incubators with sanitized. So anytime a parent or a nurse comes into contact with the infant, they'll typically sanitize their hands, or at least use the alcohol gel sanitation present in hospitals. And so because of that, we see preterms have much lower diversity than that of term infants. And we also know that the NICU has its own inherent microbiome, if you like, where we have microorganisms which exist among intensive care, and they seem particularly prolific in these populations. There's also an increased antibiotic and antifungal use. So typically, if you're born premature, you're at least under 32 weeks premature, you're given antibiotics for your first 48 hours of life until you show sign or lack of signs of any infection. And also, we have reduced breastfeeding as well in this population. So here I just highlight some of the key differences, which I'll go on to discuss in a little bit more detail. And this is just a schematic I generated for a book chapter, which is currently in press. And as I've discussed, the terms tend to have a lot more Bacteroides and also bifidol and lacto because of the receipt of breast milk, but also the increased vaginal rate in term infants also lends to more Bacteroides in this population. And overall, term infants also seem to have increased diversity and stability. So in the context of a preterm, they have reduced all of these factors, and that's important because these are all considered to be beneficial to health. So by being preterm and having these factors reduced could be detrimental to health. And coupled to that, you also have an increase in these potential pathogenic species, which include Klebsiella, Staph, Enterococcus, and Escherichia, which tend to dominate the microbiome of preterm infants early in life. But we do think that the preterm infant and full-term infant microbiome converge at some point between 1 and 3 years of life before childhood, and I'll talk a little bit about that in a moment. But before I do, I just want to touch on some recent data that we published in Frontiers in Microbiology. I think it was last week. It was published. And here we have 25 vaginal delivered infants and 21 cesarean delivered infants. The cohort was nearly 800 samples in total, underwent sequencing, so extensively longitudinal samples from each of the infants. Important to know that this was an exclusive preterm cohort, all of which received antibiotics. And between the groups, the overall demographics were fairly comparable. And then if we look then at the effect of birth mode on the microbiome profiles of these preterm neonates as they develop through time at week 1, 3, 5, 8, and then also at post-discharge, so after they leave the NICU, we actually see that there's no real effect of birth mode in this population. And so when we're looking at these type of ordination plots, for those unfamiliar with them, each of these dots effectively represents a sample. And so its entirety of its microbiome is represented by this dot. And dots that are close together, like these two, will share much greater microbiome similarities than, for example, this dot in comparison to these. And so if there was a clear difference between cesarean and vaginal delivery, we might expect all of the orange dots here and all of the blue dots here. And we could see there that the microbiome profiles were clearly distinct, but that's not what we see here. And indeed, the 95% confidence intervals really show that there's a lot of overlap between these samples. And we see that throughout all the weeks that we tested, as well as post-discharge. And then looking at some of the alpha and beta diversity metrics in consecutive samples. So the number of observed OTUs is relatively self-explanatory, and it's really equivalent to the richness of the profiles. And so each of these dots represents a sample again, and just simply the number of OTUs present in that sample. And you can see there's no difference, or no significant difference, at least between cesarean and vaginal delivery during the first 100 days of life. Nor do we see that in Shannon diversity. So with Shannon diversity, we're looking at the number of observed OTUs, as we did here, but also we're taking into account the evenness of the community. So if you have one bacteria that's very dominant in a sample, then you'll have a low diversity compared to a sample with a similar number of OTUs, but where no one OTU seems to dominate, in which case the latter sample would have a higher diversity. But regardless, we don't see any difference between the two different birth modes either. And then looking at the weighted unifrac and unweighted unifrac, again, it seems to be comparable. And just to touch quickly on what these are. So the weighted would take into account the relative abundance of the organism, and the unweighted accounts only for the presence and absence. And the unifrac element of these distance matrices account also for phylogenetic relatedness of the different taxa in the samples. So in some respects, the weighted unifrac is akin to a Bray-Curtis, if you like, but without the unifrac element. And then an unweighted would be something like a Jacquard analysis or index. And this is an algorithm that we developed for this study, and seems to be relatively novel, but was also pretty straightforward. Sorry, just get a drink. And so I'll take you through each of these points. So we have the OTUs kept, which effectively is looking at consecutive samples from the same infant. If that OTU appeared in your previous sample and your current sample, it would be considered kept. If that OTU was in your previous sample but not in the current sample, it would be considered lost. If an OTU was not present in the preceding sample but was present at some point before that, it would be considered regained. And then new OTUs gained is pretty self-explanatory. That is any OTU that is gained for the first time. And so we overall didn't see any significant differences between these, except for the OTUs kept. It does seem like vaginally delivered infants are able to keep more OTUs from one sample to the next, suggesting potentially that these have increased stability of the microbiome through time. And indeed, we've seen the same finding also in the post-discharge samples. But looking at the individual taxa, so I've talked about the 4 taxa that dominate the NICU environment, the Klebsiella, the Escherichia, Enterococcus, and Staphylococcus. We didn't see any difference, and then Bifidobacterium is usually the 5th most abundant bacteria in this population. And again, we didn't see any difference between the 2 birth modes. And so, like I stressed at the start of this little section on birth mode, that this is an exclusively preterm population, so it's different to the currently previously published evidence in term infants. And so also just to again note that all of these infants received antibiotics from birth. And so the reason that we may not be seeing any difference here is simply because we kind of reset the microbiome, if you like, and kind of override any of the potential effects of birth mode. And equally, in all the studies to date that do find a difference between the birth mode in term infants, it usually is Bacteroides that seems to be driving that difference. And preterm neonates have a stark lack of Bacteroides throughout all sampling on the NICU especially. And so it may just be that because we don't detect Bacteroides in great amounts in this cohort that we don't see the cesarean or vaginal effects of birth mode. Okay then, just moving on now to what happens to the infants after they leave the NICU. We asked for samples to be sent back from all infants who had left the NICU over a time frame, and received an okay response rate, and were able to sample roughly 30 infants following discharge. And you can see there's a huge range of ages from 14 months following discharge all the way up to 4 years of life. And we also collected their metadata from when they were on the NICU. So if they got NEC as an N, S as sepsis, or if they were just controls and didn't get NEC or sepsis. Also things like their birth weight and birth modes, and if they received breast milk or not. And actually we found no difference that I alluded to in the birth mode section, but also with things like breast milk. If you were previously breastfed, although here clearly most were, a lot of these factors didn't lead to any differences or sustained differences following discharge. And an example of this, it would be in the disease group. So we have controls, sepsis, there's late onset sepsis, loss, or NEC, necrotizing enterocolitis. And you can see during the NICU at least that this NEC, for example, seems to reduce through time. This may be some effect of having antibiotics around here, which is when you're likely to be diagnosed with NEC, and the antibiotics having this effect of reducing the overall diversity. But then I wondered if this would be sustained after they left the NICU. And as I alluded to, it doesn't appear like it is. This is just a schematic, but it gives you a sense that following NICU discharge at around 100 days or earlier, we see that the infants quickly gain a lot of diversity. And actually what was interesting as well is the diversity at this age was comparable to what it would be as a term infant. And so although these preterms clearly have a delayed establishment of diversity and very much an altered microbiome early in life compared to a term infant not housed on a NICU, they do seem to be able to catch up after they leave the NICU environment. And here's just some pictures to show that. So as I've talked about, we have these 4 core taxa which dominate the early neonatal samples and represent around 80% of that diversity by the time that they leave. So this is the last sample that we collected on the whilst they were on the NICU. It represents those 4 OTUs represent only around 50% of a clear reduction in staph. And then by the time they're in the post-discharge phase, these 4 OTUs really only represent around 10% of the overall profiles in these infants. And indeed, E. coli seem to persist relatively well, but we have this huge increase in a range of bacteria. And that's just mirrored here when we look at the Shannon diversity. You can see that the significance in the post-discharge samples is obviously way higher than that in the early NICU samples. So this really wasn't unexpected, but it was nice to at least know that the preterms could go on to develop a community diversity similar to that of terms in a very short space of time, and also that we don't see any long-lasting effect of things like antibiotics or disease diagnosis, at least in this relatively small population. And of course, we're hoping to follow this up now in a bigger cohort. Okay, then so then moving into how the microbiome influences health and disease, so away from the kind of standard development aspects. I'm particularly interested in 2 diseases, necrotizing enterocolitis and late onset sepsis. And so together, these are the leading cause of death in preterm infants. And indeed, NEC alone accounts for more deaths in preterm infants than any other pathology. The incidence of sepsis or late onset sepsis is actually higher than NEC, but we can administer antibiotics and detect this disease better, and so the mortality rate as a result of sepsis is lower, and prematurity of the infant is really the major risk factor for both of these diseases. And so that really relates to the immaturity of the intestine, as I alluded to earlier with the barrier integrity. If you as a premature infant, you'll have lower barrier integrity and tight junction formation, then you're much more at risk to a bacteria in your gut translocating across that barrier, and in the case of sepsis, getting into the bloodstream, or potentially just in the case of NEC, really aggravating that immune response. And so we see this pro-inflammatory response, or what we consider an exaggerated inflammatory response as a result of that, which ultimately leads to necrosis of the tissue. And so just a little schematic to demonstrate that here, we would expect a healthy luminal space in infants without NEC, but then when the infant started to succumb to the disease, you can see this very clear inflammation of the intestine, like I say, in its worst case leads to very severe necrosis, in which case the infant has to undergo surgery to have that section of the bowel removed. And the other thing that I'm particularly interested in, and that's why I'm talking today, because of the microbiome, is this abnormal bacterial colonization aspect. And so bacterial colonization is a prerequisite, clearly for sepsis, but also for NEC. So NEC cannot occur in utero when the gut is thought to be sterile, and nor can it occur in germ-free animal models. And so clearly something's going on with the way the bacteria colonize the gut, but also with the way the host responds to the presence of those microbes. And so a lot of my research over the last few years has been trying to untangle that question. And so really, before I get into this, I just want to highlight a recent study by Phil Tarr's group. This was a 3 NICU multi-center study, a lot of NEC cases, a lot of infants, and sequenced a couple of thousand samples. So very powerful study by all accounts. And what they found was that they binned the ages into 4 discrete categories, so 1 to 15 days of life, 16 to 30, 31 to 45, and 46 to 60. And if you compare cases and controls in the first bin, you really can't see any difference between NEC and controls. This also applies to the second time point, but then following day 30 of life, you can actually see a huge increase in this gamma proteobacteria in infants that developed NEC compared to infants that did not. And so this group only sampled pre-NEC cases. So once they were diagnosed with NEC, they were excluded from the study. So this only includes pre-NEC samples. And so this does seem to be, and what I noted here is after day 30 of life, seems to be diagnosed in a very specific type of NEC. Most NEC is diagnosed prior to day 30 of life. Even in this study, I think the average age was 30 days. So certainly most were diagnosed by that point. And in the UK centre that I collaborate with, we typically see the average post-gestational age of NEC or day of life of NEC to be roughly day 20. So a NEC occurring after day 31 does seem to be a relatively exceptional NEC, but nonetheless, these authors were able to see microbiome differences between those later NEC cases. And equally, the same sort of thing with the Shannon diversity. So early on, the cases and controls were very comparable, and for us, that wouldn't allow us to represent or detect differences in the most of the NEC infants, but then certainly in these later NEC cases, you can see that the controls go on to have much greater diversity than that of the NEC infants. Clearly, there's a huge range within the controls, but that also is not to be unexpected in this very vulnerable population. And one thing else that the authors note was just that these findings were driven by infants typically under 27 weeks gestation, which is, as I mentioned, NEC is exclusively premature disease and also inversely correlates with the gestational age. So you're at bigger risk the earlier in your gestation that you're delivered. So it's not exactly surprising that these findings were driven by infants under 27 weeks. And then on the back of a lot of animal work that was being released around the time, again last year, it was reported that it wasn't so much about the which exact bacteria or taxa or species that you had in the gut, it was more about the total bacterial load. And so we explored this in our clinical cohort. So 2 weeks prior to NEC, 1 week prior to at diagnosis, and then 1 week and 2 weeks after, we compared NEC samples to control samples, looking at the overall bacterial load in these samples, and we found no differences that would allow us to use this as a biomarker for NEC development or even to understand better the mechanism of NEC. The only difference that we did see was in NEC cases 1 week after diagnosis, by which point a lot of antibiotics would have been administered, and it's not surprising that we see the amount of bacteria in NEC cases drop significantly, and this was somewhat withheld even in the 2 week after NEC category, but this didn't meet significance between cases and controls at that point. So really there didn't seem to be a significant difference between the bacterial load, at least in the clinical cohort of NEC. So we took this further and looked at sequencing samples using, as I've discussed, the 16S approach, particularly targeting the V4 region. So this was initially one large study, and then through reviewer request, we've split this into 2 manuscripts. So we have 28 control infants and only 7 NEC and 7 loss. We had relatively strict exclusion criteria for this study, so you had to have extensive longitudinal sampling both before and after disease to be included, and also NEC is inherently a rare disease, and so that's one of the reasons that the N is relatively low. So we collected, like I say, extensive longitudinal samples from these infants, and overall demographics are relatively comparable, except for clearly we'd have a lot more antibiotic administration in the diseased cases. And so the next study is now published to Microbiome, and the sepsis study has been accepted to Microbiome and it is currently in press. And I'll talk you through some of the data in those papers now. And so here's just an example of 2 patients, 117 and 131. And again, the 4 core OTUs, which I've talked about throughout the study, are shown here in the bottom grid. And then just because we couldn't plot the satellite or lower abundant OTUs together with these, because they would really just appear as a baseline, these are separate, plotted separately with a different scale here on the axes. And so just to think about how a normal, or what we would consider healthy preterm infant develops through time, one may expect that you would see some signatures that would allow you to really predict what a healthy microbiome looks like. But at least visually, based on these type of plots, really what I want to try and do is highlight how sporadic the microbiome development appears to be within this population, both between infants, where there's clearly a vast amount of differences, but also within infants. So we can take many examples. For example, patient 143, where at 2 weeks of life, they were dominated by staph. Then at 3 weeks of life, they were dominated by enterococcus. And then at 4 weeks of life, they were dominated by Escherichia. And so clearly there's a huge dynamic development of the microbiome, even within individuals. And so that makes it very difficult to try and then ascertain why some infants may go on to develop NEC when we can't even define necessarily what a control population should look like. And again, this really just stresses the importance of doing these extensive longitudinal studies, because to pick a sample at any one of these time points and consider it as a cross-sectional sample clearly wouldn't be very indicative of what the microbiome truly looks like, because it could literally change within 24 hours in this population. And so here is just area plots of the NEC and sepsis profiles. So each of the infants that succumb to NEC are plotted here on the left, and the red line represents the day at which NEC was diagnosed. And the same for the sepsis infants, only here the blue line represents the day at which sepsis was diagnosed. And you can see there doesn't seem to be any one taxa which really strongly persists throughout the NEC population. And just visually looking at this, you can see that the changes in the community are very individualized. Indeed, some infants don't seem to have any changes and still go on to develop NEC. And so it was very difficult, again, to map a specific taxa that's causative to these diseases based on these data. One thing to note within the sepsis population, at least, where we can see here the blue line represents diagnosis, but the text next to it represents the organism cultured from the blood, is that we see the dominant organism at the time of diagnosis was typically what was also cultured in the blood. And so here staph would be represented by the purple and is clearly very dominant, and that was also always isolated in the blood. Enterococcus here in blue. And then we also see cases like this, such as the group-based strep, where it seems to really sporadically appear, and the infant succumbs to the sepsis, and then the antibiotics are administered and it drops off. But this is in line with some data from other research groups that look at the strain level and can show that the exact same strains which dominate in the gut are also the ones that are isolated in the blood culture. We can't go to strain level with 16S data, of course, but other data supports these types of findings. And so it does lend itself to the potential that we could, in a much quicker time frame than pathology could do of routine blood culture analysis of these sepsis infants, we could potentially sequence the gut microbiome and allow clinicians to tailor antibiotics accordingly at what seems to be dominant in the gut. Two interesting cases emerge out of this study. One was diagnosed with NEC earlier on in life, but then died of NEC only shortly after diagnosis. Another infant was only ever diagnosed with sepsis, but then post-mortem most likely also had NEC. And you'll notice that in the last sample collected from each of these infants, there was a huge colonization by this Escherichia coli. So it was intriguing. These infants, as you can see by the numbers here, the patient numbers 178 and 180 both existed on the NICU at the same time. And so I did wonder if this was a single kind of golden bullet of E. coli that seemed to be causing NEC in this population, or indeed potentially contributing to the NEC pathogenesis in this population. So we did some pulse field gel electrophoresis of these samples, and actually we didn't find any similarity between the two strains that were isolated from the disease cases compared to that of controls that also had a lot, or also had dominance of E. coli but never succumbed to disease. And so every infant seems to have its own very unique strain, which again is something that's now being widely reported as the methods are getting more and more sensitive, and we can go to a deeper resolution levels using things like metagenomic and whole genome sequencing. And so there doesn't seem to be a single taxa that causes NEC, and this really is in line with a lot of studies. So some studies, such as Phil Group, can highlight some phylum or classes or families of bacteria that seem to be associated with the disease. But for every case where that seems to be the case, there'll be many other cases where they didn't seem to potentially get those taxa. And so to explore this a little bit further, I did some clustering using PAM and defined 6 preterm gut community types as highlighted here. And so you can see Klebsiella oxytosa dominates preterm gut community type 1. Both Klebsiella and Enterococcus were dominant in group 2. Staph is dominant in group 3. Enterococcus only in group 4. E. coli dominates group 5. And then group 6 is hallmarked by a dominance of Bifidobacterium. And equally, group 6 also had the highest diversity overall, and also the highest number of OTUs overall. So then when we consider those using Markov chain analysis, we can first of all just highlight group 6 here, which was the high diversity Bifidobacterium rich group. You can see that their fraction of NEC is 0, or at least in terms of their pre-NEC samples, saying that if you have group 6, you never went on to develop NEC. And like I've kind of just touched on in the previous slide, we don't see any of these preterm gut community types associated with the development of NEC. All of these were relatively low. So it does seem like there isn't a specific causative agent to NEC in this case, but at least a community type which was reflective of community type 6 would seem to be protective from the disease. And indeed, we also seen the exact same findings in the sepsis cohort here. If you were colonized by community state type 6, then you had a 0 fraction of sepsis, and also we've seen that in group 2 as well, which we didn't see with the NEC infants. And then just monitoring these through time just to allow you to visualize this in a slightly different way. Here you can see the diagnoses as they were on the previous slide, with red representing NEC and blue representing sepsis. And indeed, the only case of preterm gut community type 6 represented by the pink dot that occurred in any of the disease samples occurred in patient 199, who also, I must note, had NEC diagnosed much earlier in life, and so by this point would be considered relatively healthy. Equally, preterm gut community type 6 occurred throughout the control population, even quite often in early samples. And so it's intriguing to think that colonization by not only Bifidobacterium, as that was the most abundant taxa, but also this community that's also highly diverse may allow protection from necrotizing enterocolitis. And so one of the other things that we noticed in this study was that the transition from one community type to the next seemed to influence the risk of developing NEC. And so NEC infants seem to show much more chaotic development, bouncing between community state types a lot more than, say, controls who only seem to go through 1 or 2 state types during their development. And here's a video just to highlight that. And so you can see the red line will be a neck infant who moves very quickly through time for this ordination, going through a range of community state types, whereas the control infant shows much more similarity, moving from sample to sample, suggesting that their microbiome profiles are not changing very much through development, only very slightly. You'll also notice that the control infant spends a lot of time in community type 6, which, as I mentioned, is that community type that's dominant in Bifidobacterium. And so you can just get a sense now, just looking at the plot, after showing that development, that the both started from a very similar space. They both started from community state type 3, but very quickly the neck infant was moving all around various different state types, moving quite a long way across this ordination plot, suggesting it's moving from very distinct state types and completely separate state types, whereas the control infant tended to not move as far through this ordination space, suggesting its community remained much more stable. And then we also did some metabolomics. This was fairly pilot in nature, and using metabolomics, LCMS-based metabolomics on a subset of the stool samples, we were quite confidently able to differentiate neck from controls. As you can see here, it's not a great number of patients or samples, but we did highlight several pathways, and linolate metabolism came out on a couple of occasions here in some of the modeling that we did. And as you can see here, the T3, which I'm highlighting in red, represents the date at which that neck was diagnosed, compared to the blue square, which represents the equivalent sample in a control infant. And you can see that as neck development increases, you can see this increase in this leonate metabolism metabolite. And by the time at which neck's diagnosed is also when it's at its highest. And then following diagnosis and treatment, you can see this metabolites start to drop out. So like I say, very pilot in nature, but does lend itself to potentially further exploration as a biomarker, or at least allowing us to understand better the mechanism of necrotizing enterocolitis. And then also by doing some, and this will be my last data slide before I go into a summary, but just to quickly know that we also can do some correlation analysis of these. And here you can see that the Bifidobacterium is strongly correlated with things like raffinosis. One thing I wanted to particularly point out is this is a metabolite which is thought to be beneficial to host health to have, and is reflective of a healthy gut ecosystem. And so it's interesting that these taxa do seem to be directly correlated with beneficial metabolites and an overall beneficial function. Okay, then just to leave plenty of time for questions, I'll wrap up my summary. So longitudinal 16S or RNA gene studies are very useful, and they give us a good survey of a community, but often it's limited to non-invasive sampling. So in the cases I presented here, it's limited to non-invasive stool sampling, and we regard stool sampling really to reflect a distal colon, whereas when we consider diseases like necrotizing enterocolitis, which most typically occur in the terminal ileum, perhaps stool sampling isn't the most representative sampling for that. The application of additional omics technologies, which I really only touched on at the end there in the interest, this is a microbiome specific talk, so I stuck to microbiome, but a lot of this data is published, so you can feel free to go away and read about that and get in contact if you have any further questions regarding some of the multi-omic analyses. But by applying these different omic technologies, such as transcriptomics, proteomics, and metabolomics, it allows us to begin to get a picture for not only what bacteria are there, but what they're doing, how they're interacting with the host, and how the host is responding to the presence of these microbes. And just to note that correlations are important, but really this doesn't tell us anything about the causality. And so what we really need to do after we do this discovery-based research is require, as I outlined earlier in the talk, it requires us to further validate in animal models or ex vivo cell culture models. And that's all well and good for a lot of studies where there is a clear animal model for going away and testing your microbe or metabolite of interest to see if you can restore the disease phenotype. But in a preterm population where animal models don't necessarily well represent what a preterm immature gut looks like, if we use pigs, we typically deliver them 7 days early, or in mice models, it's 1 day early. That really isn't representative of a 23, 24, 25 week gestation infant. But one area, as I touched on earlier, again, it is the enteroids, which I think is going to be super exciting. It's something that I'm increasingly working in now in the next space to try and derive really viable models of prematurity, which will allow us to interrogate the mechanisms of things like necrotizing enterocolitis and hopefully develop new therapeutics for preventing the disease in the first place. And then the other thing, there's a recent publication regarding organoids in neonatal and preterm research, recently published as a preprint, which I would encourage people to read. Again, this may pave new frontiers in preterm research, and ultimately allow us to combine in such a co-culture fashion where we can look at the response of the primary human cells to the microbiome and also to the immune response. And so working over the last 10 years in this space, I've had the fantastic opportunity to work with a range of really awesome people. For a lot of the work I've presented today, it's all been based on Janet Barrington and Nick Embleton's real passion for collection of preterm samples and doing these types of studies to hopefully improve long-term health outcomes for the neonates. So continue to collaborate with those and really appreciate their input throughout. Also, Professor Stephen Cummins is my former PI and worked heavily in this area, and Andrew Nelson, a postdoc at Northumbria, also assisted me a lot in my early days of doing this work. And now currently at Baylor College of Medicine, I'm very fortunate to work with some fantastic minds, Joseph Petrosino, our PI, also Daniel Smith, who helped with a lot of the bioinformatic analysis, and Tatiana, who's helping me now with a lot of the enteroid analyses. And then some other groups about research that I didn't get to talk about, including the respiratory microbiome, which I wanted to, but in the interest of time and leaving time for questions, I decided to omit that. But again, more than happy to talk about that kind of work, and indeed what we just recently published in the Blue Journal, a paper in this area. So if you want to read it and have any questions, again, feel free to read that and get in touch with me. And to get in touch with me, here's my email address. Feel free to follow me on Twitter. And also Nick Embleton of the RVI has done a fantastic job setting up this website where he regularly updates and blogs about not only our research, but other research in this space. And then just to touch on the QIAGEN, or formerly Mobile, now QIAGEN Microbiome Award. As I said, I'm here today because I won this award a couple of years ago, and I would encourage anyone who's thinking about applying to apply. It's really a fantastic way to just get some preliminary funds to do an experiment that you're interested in doing, but also allows you to have a little bit more exposure in the microbiome space, which is really always advantageous when you come to looking for jobs and funding in the future. So I'll leave that there. I'm happy to open it up to questions. And again, once again, thank you all for joining in and for listening today. It's much appreciated.
Thank you, Chris, for a wonderful webinar. Thanks again. And we have received a lot of interesting questions from the audience, and now I think we will take one by one. So the first question is basically, how reliable is this comprehensive 16S rRNA?
Sorry, was that how comprehensive is it?
No, how reliable is comprehensive 16S RNA?
Again, I'm sorry, I'm just going to get you to repeat that one more time. Is it how reliable is 16S RNA?
How reliable is comprehensive 16S RNA data, basically? I think
okay, well,
the audience, yeah.
Okay, if I think I understand the question correctly, 16S RNA sequencing is, I guess, touched on, allows us to survey the community very well. For me, it still comes into very much discovery, and it allows us to get a much better understanding very quickly of a community, such as in a stool sample, which would otherwise be very laborious using, for example, microbiology-based techniques and culturing out organisms. But certainly as the technology evolves, a lot of microbiome has really been driven by advances in technology, and that's not likely to change anytime soon, as the ability for us to perform either longer sequencing reads to get a much better idea of the 16S and hopefully identify to species level, or even better to now bring down the cost of WGS sequencing and really start to apply metagenomics. I think in maybe 5-10 years, most researchers, as we initially moved in the last 5-10 years from gels to sequencing the 16S RNA gene, I think we'll move from that now into just doing community-wide sequencing for which will also give us species level identification, and then also allow us to get an idea of metabolic potential of the community also.
Okay, thank you. The next question is, microbiome data is primarily associative. How do you know microbiome diversity is related to outcome? So what are the practical steps to determine mechanistic relationship?
Yeah, that's a fantastic question. It's really the crux of microbiome, is moving beyond these associations, as I kind of alluded to just there in the summary. There's a range of diseases where we know that the diversity is reduced, and therefore that's really the basis for a lot of our kind of potentially oversimplistic statements like diversity. Low diversity is bad for health, and high diversity is good for health. Certainly in most ecosystems throughout time, essentially, in evolution, like a diverse population is usually for the benefit of all. And there's several reasons why having a high diversity microbiome would be beneficial. But yeah, for sure, a lot of this is association-based evidence, and a lot more systematic and kind of basic science is needed away from clinical samples to really get at if truly diversity is a reflection of a disease state, or if it's just an effect of a disease state already occurring.
Thank you, Chris. And the next question is, what is your opinion about the formation of the microbiome in the prenatal period in relation to the developmental origin of health and disease theory?
Yeah, so again, I guess I touched on in the talk, I feel like this is a fascinating area of research. I really don't think we're there yet in understanding too much about the prenatal exposure to the microbiome. It is possible if DNA can cross the placenta and colonize, or not colonize if it's DNA, but get into the fetus as it's developing, that it could elicit some immune training. But honestly, it's definitely where that we're going to go in the next 5 years to begin to understand this, but we're just not there yet. I think potentially it has very important connotations, but it definitely requires further work. It's probably the most contentious microbiome area at the moment, I'd say.
And okay, so for neck, do you think that specific gut microbiota are less important, and the translocation of any bacteria is the important driver of neck?
Yeah, and I think that's a very nice point, actually, a nice way to summarize a lot of what I've said. And yeah, for sure, I'd agree with that. I think it doesn't seem to be about which specific taxa that you have. Certainly not any particular species or genre. Every infant seems to have a very unique development, and there doesn't seem to be any one taxa which consistently falls out. Even within a single NICU, like I talked about a lot today, the microbiome is clearly very different between neck cases. If you then consider comparing that to different NICUs with different exposures and practices, it gets very convoluted, but the overall story is that there isn't one single bacteria that seems to be consistently implemented in neck. So it may not necessarily be so much about which potential pathogens, if you like, that you have, but more about trying to administer this healthy community, and a hallmark of that in our cohort was what filtered into community state type 6, which, like I said, was high diversity, but also very dominant in things like bifidobacterium. And the good thing about bifidobacterium is there is a lot of basic evidence as to why this may be beneficial for a neonate. And one thing that I always relate to preterm infants is that bifidobacterium is known to stimulate the maturation of the gut epithelial layer, and so it can promote tight junction integrity and barrier formation. And so having a lot of that species promoting those sort of features in the host would clearly have huge connotations for a preterm infant, where I think a lot of the reason that the exaggerated inflammatory response begins in the first place is just because they have this immature and leaky gut to begin with.
Okay, thank you, Chris. And the next question coming from the audience is, how does intervention with lacto-bifido or both probiotics alter the neck outcome?
A great question, and again, this is a fascinating area that we're only beginning to get a sense of. There has been a lot of small probiotic trials and some meta-analyses of those, which suggested that overall probiotics seem to reduce the rate of neck. Typically, as the audience member asked, it's primarily through lactobacillus and bifido are the primary probiotic candidates in this population. But then recently, also the largest probiotic study anywhere was performed in the UK, and they used a single strain of bifidobacterium, a bifido breve strain, and they didn't see an improvement in the neck rate. And so it definitely lends itself to further research. It seems like we really don't understand which is the best strains to be given, and it's very possible, if not highly likely, that each individual probably needs slightly personalized a probiotic. So if they're getting breast milk, for example, things like bifido breve would probably do well because of the HMOs, but other things like lactobacillus may not. And so we may need to begin to start to think more about these probiotics rather than just assuming that putting a good organism in a baby is going to do good. We really need to think more about other factors, what it's being fed, is it getting antibiotics, what's likely to survive and colonize, and these sorts of things. In some preliminary data from us in the probiotic space, we published this a couple of years ago now, but we found that bifidobacterium does go in and colonize and seems to stick even after discharge, whereas other species like lactobacillus bacillus we only detect during being probiotic, during a probiotic administration. And so I suspect that what's happening there is the probiotic is really just flushing through the gut, which isn't to say it's not having an effect while it's doing that, but it's certainly not colonizing and talking to the host in the same way that bifidobacterium would be.
Thank you, Chris. And maybe the last question, because we are already running out of time. So is there huge variability in minor taxa you observed in the preterm as well as the neck infants, similar to what you would see in term infants in the early days of life?
I'm sorry, can you just repeat that, please?
Yeah. Is there huge variability in minor taxa you observed in the preterm and neck infants, similar to what you would see in term infants in the early days of life?
Yeah, again, a good question, and it is. So in the early days of life, well, as I discussed with the birth mode stuff, your microbiome very much reflects your birth mode immediately following delivery. So if you were born by vaginal delivery, you tend to have a lot more lactobacillus, which are typically dominant in the vaginal tract. And if you're delivered by cesarean section, then you'll have a lot more maternal skin organisms as well as environmental exposures, environmental organisms. And so those organisms are very much dependent on what the mother is colonized by and what organisms persist in the environment. And so there's a range of factors why any one individual would be fairly individual. However, despite this individuality, there is definitely common trends, as I discussed those two papers from Science Translational Medicine in a current analysis and performing of over 10,000 samples from over 1,000 term infants. We definitely see the same kind of patterns in the development of the microbiome. So while everyone is individual, everyone definitely kind of follows the same overall trajectory, but there are certain variables such as breastfeeding and birth mode which definitely influence them to some degree.
Thank you, Chris, for a wonderful webinar, and thank you everyone today for joining this webinar. And we are looking forward to see you again in next webinars from QIAGEN. And I wish you all a good start or rest of the day. Thank you so much. Bye-bye.
QIAGEN.
Sample to insight.